Let's look at the structure of a particular protein kinase B, also known as AKT. Protein kinases, there are 518 in the human genome. They key proteins because they phosphorylate other proteins and in the process turn on and turn off metabolic and signal transduction pathways. In addition, they themselves must be phosphorylated or dephosphorylated or somehow regulated, so their activity is turned on and off as well. Here's a generic active protein kinase cartoon. All protein kinases must bind ATP in the active site and also a target protein for phosphorylation on a serine, threonine, or tyrosine side chain. They typically consist of an N lobe in the N-terminal region, a C lobe in the C-terminal region. The N lobe also consists of 5 beta strands and 1 alpha helix. This helix is called the C helix, unfortunately. In the C lobe, there are 6 alpha helices. Note there's some also key loops. There's the activation loop shown in blue and the catalytic loop shown in red. Now, this activation loop has shown here is phosphorylated on threonine 197. This is the active conformation of the enzyme. Typically, the activation loop is intrinsically disordered, and it's essentially blocks access to the active site or reduces access to the active site, and also the key catalytic residues involved in catalysis are not in the optimal position. On phosphorylation of the activation loop, it adopts a more localized conformation, not so disordered, doesn't obstruct the active site, allowing ATP and the protein substrate to interact. And in this process also repositions the key catalytic residue so catalysis can occur. So again, this is the active kinase. Almost all active kinases have the same sort of numbering system for the key residues and activation loops. What we're going to be looking at is AKT. It has in addition an N-terminal peak, a domain called the plextrin homology domain, pH. And so the numbering system is slightly offset, but we're going to be modeling this protein today because it's so many things to model. The key catalytic residue is the activation loop, the catalytic loop, and the N lobe and C lobe. As we said, AKT is activated by phosphorylation. It does so by being recruited to the membrane where it can bind through its plextrin homology pH domain to an inositol lipid PIP3. This derives from PIP2 by phosphorylation. The structure of PIP3 here is shown here. So this AKT is recruited to the membrane. In addition, PIP3 recruits the kinase that will phosphorylate AKT in the activation loop to activate it. That kinase is PDK1. So when it phosphorylates threonine 308, and in addition, another kinase which has been activated by PIP3 phosphorylates a serine 473, now we have AKT that's fully phosphorylated. The activation loop now has this more localized conformation that allows key residues in the active site to encourage catalysis and also allow substrate binding. Given the importance of this protein, no wonder that there's a lot of PKB structures for it. We're going to concentrate on 3 today in the modeling. And this is sort of a complicated diagram showing the kinase in the cytoplasm free and also membrane bound. Now notice that the orange domain is the pH domain. In this case, the yellow structure is the full kinase with the N and C terminal lobes. Like there's some circles for connecting these lobes. In those cases, those are sort of cartoon connections because the crystal structure is not known. But if you look at overall the structure, there's this sort of appendage coming out here and here and here. These represent the inactive forms of the kinase. When it's phosphorylated at the key site, we have a conformational change, and now we have the active protein that's represented by this. That particular PDB is 3CQW. The inactive form is 3096. And in addition, there is another PDB file called 1UNQ in which it's actually interacting with the target membrane. So we're going to be looking at each one of these structures today using ICN 3D. Before you do the modeling of ICN 3D, you should go to Uniprot, which is one of the most amazing websites for information on proteins. Here's the URL for this particular protein, AKT1 in humans. In this page, you will find information on post-translational modifications, all the sites that are post-translationally modified. In addition, you'll find information on families and domains. It's a really robust site. If you go to that, within Uniprot, you can find information from the PFAM and the Pro site, and these give cartoon representations, sequence representations of the domain structures. So for instance, here is the pH domain of the AKT and the kinase domain and an additional secondary domain. The same sites are found by another program that actually looks at domain structure. So in summary, we're going to be modeling AKT. It's a splendid model, molecule to model, because it has such interesting features. You'll be modeling the N lobe, the C lobe, the activation loop, the catalytic loop, and actually the membrane binding domain as well.